So I would like to congratulate these organizing members of this workshop. Yes, yeah, the organizing members of this workshop, and I would like to congratulate you also, so patiently listening the talks from morning to evening and actively participating in the sessions, tutorials, as well as all these things. And today I'm going to talk a very small topic, I think related to a very important component, call them as a phenomena, call them as a acclimation. So what do you mean by acclimation? What do you mean by acclimation? Suppose you travel from very distance to University of Hyderabad. Yes, the moment once you reach here, I think you feel some discomfortness. Yes or no? But after 10 days and we are about to finish this workshop, now you feel comfortable. Yes or no? Yes? Yeah. So means you have adapted. Means we have adapted or we have adjusted the specific environment. A specific environment. So this phenomena is always called it as a acclimation. Okay? Acclimation. Then here the word is called as photo-acclimation. What do you mean by photo? Photo means light. Okay? So because light is the major source of energy, and according to the first law of thermodynamics, in the process of photosynthesis, the light energy is converting into chemical energy. Light energy is converting into chemical energy. But the amount of energy which is present in the universe is equal. Okay. So, but the energy can be converting into one energy form into the another energy form. So this is called as that is energy neither be created nor be destroyed, but it can be converting into one form of energy into the another form of energy. So this is called as a first law of thermodynamics. Now, so I would like to ask one important question here, because I'm going to talk this concept regarding about this climate change. I think everybody is talking about climate change. So I would like to ask one question here. Is climate change real or not? Our climate is changing? I would say no. What is your answer? So how do you say that our climate is changing very rapidly? Only flora and fauna? Why flora and fauna is changing? This is the science of climate change. So here, I think intense drought episodes. We are experiencing heat waves, rising sea levels, melting glaciers, warming oceans. So these all are the different signs of climate change. Then what are the factors which are driving the climate change? The most important factors which are driving the climate change is the emissions of the unprecedented increase in the greenhouse gas emissions. I think you all know that, I think in the form of modernization, in the form of civilization. So the human beings are moving from these sustainable practices into the unsustainable practices. So when we are trying to make more unsustainable practices, so we are releasing more greenhouse gases into the atmosphere. So the moment the threshold levels of greenhouse gases are increasing, so which create lot of problems to society as well as our environment. So I think you all know that, I think so nowadays I think everybody is facing lot of health complications like bronchitis, cancers, different types of diseases and all this. Because of all these are most likely to be integrated with the climate changes. Now, see, I think you just see this, I think this figure, I think among all greenhouse gases, the carbon dioxide contributes major proportion. I think see here, I think around nearly 76%, followed by the methane, followed by the nitrous oxide, as well as followed by the fluorochlorocarbon gases here. So these all are emitting into the atmosphere, and which are causing hell of problems to the human individual as well as our climate, as well as the environment. Now, another question is, does CO2 rise follows temperature rise? I think you just see here, I think this graph clearly indicates that, I think these are the CO2 levels before the industrial revolution or modern human modernization or civilization. I think there were, I think you know very well, the CO2 concentration used to always, I think they are measured in the form of ppm or micromole. What do you mean by ppm? Parts per million. Parts per million. Okay, now see here, before industrial revolution, I think what's happening here, I think in the year 1880, I think, so what's happening here, I think the CO2 levels were around 270 parts per million, but due to modernization and civilization, so the CO2 levels are continuously rising, and now I think in the year 2019, it got reached around nearly 410, now the CO2 levels would be around 420 ppm. So you just imagine that in a span of nearly 100 years, I think there was a significant rise in CO2 levels, So I think you must have feel very hot nowadays because of this rise in CO2 levels. Now, see here, at present, I think the CO2 level would, I think, is around 420 ppm. Again, so if you won't take any preventive measurement, if you are not conscious, what's happening here, I think by the year 2050, I think it would be reaching around nearly 550, and by the 2100 it would be reaching nearly around 700 ppm here. So you just imagine that the moment once it reached to CO2 levels would be reaching around nearly the 700 ppm, I think there must be rise in average global temperature by means of this I think 3 degrees centigrade here, 3 degree centigrade here. So you just imagine the severity of the climate change. So when I was working at University of Hyderabad, so the CO2 levels would be around I think 400 and around 395 or 380. Now you just imagine, in a matter of I think 10 years, it got changed nearly, I think it is reached nearly around 420 ppm. So you just imagine that every year there would be rise in nearly 2 micromole of CO2 is increasing without, I think, so this is the clear symptoms regarding about how we are going to face a lot of complications of the climate change. Now, so how to mitigate this increasing atmospheric CO2? So what could be the major root cause of CO2 rising? First one is deforestation. Yes or no? Then second one is extensive usage of fossil fuels. So if you reversing these two processes, definitely we would be able to normalize the CO2 levels. Yes or no? Yeah. So how do you normalize the CO2 levels? So because we have to practice afforestation. What do you mean by afforestation here? Creation of artificial forest. So the artificial forests are also called as agroforestries. Agroforestries are the short rotation forestries. So what are short rotation forestries here? Suppose by you find very huge area which is not using for farming, or you find some degraded land which is not fertile at all. So you just select that area. That area can be used to cultivate the fast growing tree species, and this tree species which will go over a period of time, and you just harvest the biomass, and this biomass can be used to generate bioenergy, and in the form of bioethanol or whatever it may be here. So this ethanol can be acts as a fuel. Okay, so we are partially replacing the fossil fuels. By using this biofuels. So for this purpose, we need more amount of biomass. So that's why I think we need to efficiently utilize this. The unutilized lands or unfarming land. So that we could able to continuously generate the biomass, and this biomass can be used to produce the different types of energies here. Including bioethanol here. The bioenergy, that is the energy which could be generated through, I think, this component, I think, call them as a carbon neutral. Why? Because, so the amount of CO2 which is released into the atmosphere, again, which will be taken up by these agroforestry here. You just imagine here, so here, these are the short rotation forestry. We just cut down the biomass. And so what's happening here? So this biomass can be used to generate the bioenergy. Now this bioenergy, again, it will release the carbon dioxide. No, again this carbon dioxide will be sequestered by the same tree species again. So this process will continue over the 40 to 50 years. Now what could be here? So this type of bioenergy is always called as a carbon neutral bioenergy. Why do you call it carbon neutral bioenergy here? The amount of CO2 which is releasing is equals to the amount of sequestration here. So this is called as a carbon neutral bioenergy here. Now, I think this is a well-known phenomena, well-known biochemical reactions, I think you have been seeing from the day one from this workshop. Yes or no? So I think is the photosynthesis, I think the basis of life on Earth. Without this photosynthesis, there is no life on Earth. Yes or no? So which is mostly comprises by two different biochemical process. The one is a light reaction and another one is dark reaction. So what is the use of light reactions here? I think the light reactions are always meant to produce ATP molecule as well as NADPH molecule, because this can be used by this I think dark reactions, I think Calvin-Benson-Basham cycle, so to sequester the carbon dioxide here. So what are the end products of the Calvin cycle here? Either sucrose or starch molecule. Yes or no? So here again, so the Calvin cycle always comprises two different biochemical process. One is RUBP carboxylation as well as RUBP regeneration. So RUBP carboxylation can be catalyzed by the most important enzyme called them as Rubisco, the most abundant protein. Then again the another topic called them as RUBP regeneration. RUBP regeneration, I think so which can be catalyzed by so many biochemical reactions, so many enzymes, but among those I think simple three major regulatory enzymes are there. What are they? Chloroplast, pseudoepulos 1,7 bisphosphatase, fructose 1,6 bisphosphatase, and phosphoribullokinase, and three are the major regulatory enzyme. Why I call as a regulatory enzyme here? Because these enzymes will always control the flux of carbon dioxide into the Calvin cycle. So that's why I think these enzymes are always called them as a metabolic valves, which will control the flow of carbon into the pathway, so and which could eventually affect this I think the growth of the different consequences here. Now, so why do you focus more on this Calvin cycle is because people try to manipulate the enzymes of Calvin cycle and the proteins of this light harvesting that is light reaction, so that people could observe some enhancement in the photosynthetic efficiency, which eventually resulting into increase in crop productivity here. So that's why I think we mostly likely focusing on this photosynthesis, especially I think light reactions and dark reactions here. So in order to perform efficient photosynthesis, I think these two processes needs to be coordinated with each other. So without light reaction, there is no ATP and ADP. Without NADP, ATP molecule, there would be no proper activity of Calvin cycle enzyme. So that's why for a better photosynthetic efficiency, these two processes needs to be coordinated with each other, and without coordination, I think there would be no proper photosynthesis here. Now here, so how to suppose you have four different plants with you, okay? Plant A, plant B, plant C, plant D, okay? So you wish to know which plant is performing better. Photosynthesis. So in order to track the photosynthetic changes, we have to track light reactions, we have to track the changes in dark reaction, especially Calvin cycle. So how to track this light reactions, how to track the changes in Calvin cycle, dark reaction? So I think you have seen this instrument, I think in SAR's lab, I think you have seen this instrument, that is handy P. So the handy P will always majorly focus the changes in the efficiency of photosystem 2, that is the flow of electrons from the photosystem 2 to photosystem 1. So which majorly emphasizes, I think whenever we are using the handy P, we could able to see the four different parameters. I think Fe by Fm, call them as a maximum quantum yield, followed by the performance index, electron transport, and dissipation. So these all will always emphasize majorly, I think the photochemical quenching as well as non-photochemical quenching. Later we will discuss. Okay, then I think you also see this instrument, IRGA. Yes or no? What do you mean by IRGA here? Infrared gas analyzer. So this instrument will always be able to emphasize the changes in the Calvin cycle, efficiency here. So by using this IRGA, I think we could able to quantify changes in photosynthetic efficiency, water use efficiency, transpiration rates, stomatal conductance, as well as the intercellular CO2 concentration. So what do you mean by what is intercellular CO2 concentration here? I think you know very well. The moment the CO2 will enter into the plant cell, so which will concentrate inside the chloroplast. Yes or no? So I think suppose there is a what I told here, 420 ppm CO2 is there in the atmosphere. The moment once it passed through the stomata, so what would be here? So the intercellular CO2 concentration would be around, I think 3/4 of the atmospheric CO2 concentration here. So we are seeing now 420. So I see intercellular CO2 could be around 300 or 340 like that here. Yes or no? So here, this could be a way, I think, how to track this changes in the, I think, photo system to, I think, photo efficiency here. Now, by using this IRGA also, again, we can also track, we can also assess the in vivo carboxylation efficiency of Rubisco. In vivo carboxylation efficiency of Rubisco by performing a simple experiment known as A by Ci curves. A by Ci curves. See here, what is happening here? Here, photosynthetic rates, and here the CO2 concentration. So, with that instrument, what you do that, if you provide the artificial CO2 to the plant by program, in this program, we will increase the CO2 concentration from 0 to 1000. So, what is happening here? Suppose we just fix a program in such a way that, so the program runs for 30 minutes. Okay. So, what is happening that here, we just keep the leaf and hold it inside the IRGA and keep it for that with tripod, whatever it may be here. And so, we will fix the program in such a way that the CO2 levels are increasing gradually from 0, 100, 200, 400, 600, 800, 1000. Okay, so the moment once we keep on gradually increasing the CO2 concentration, so what's happening here? So there is a significant rise in photosynthetic efficiency at certain level. After that, I think there would be the appearance of saturation here. So here, now this typical ideal, which one regarding about this A by Ci curves can be divided 3 different regions. One is which one? I think this is this region will always emphasize this Rubisco efficiency. RUBP carboxylation efficiency. So up to this 400 here, up to this 400 CO2 levels here. Then again up to 700, I think this region will always emphasize the efficiency of RUBP regeneration. Again, next one followed by the triose phosphate utilization here. So what is happening here? Suppose if you rise in amplitude of this graph at this region, which indicates that there is an increase in activity of Rubisco. Suppose if there is a rise in amplitude of the graph around RUBP, which indicates that there is an increase in RUBP regeneration capacity. So this will clearly emphasize that, I think what's happening that here, so which will analyze this, which will we will come to know that, so which plant is more efficient, by performing this simple A by Ci curves here, I told you, see here, I think Rubisco carboxyl efficiency can be controlled by 3 different enzymes. What are they? Rubisco activase and carbonic anhydrase. Again, next one, RUBP regeneration can be controlled by, I think, different types of enzymes. What are they? Pseudoheptulose 1,7 bisphosphatase, fructose 1,6 bisphosphatase, and phosphoribullokinase. So these are the 3 different enzymes which could always be able to contribute for the efficient RUBP regeneration here. Now, this is another type of measurement we could use always to track this photorespiration. So how do you track the photorespiration here? I think so here, this is called as a, I think CO2 assimilation rates versus increasing photon flux density. So what is happening here? Similar with the A by Ci curves. So there was, in the A by Ci curves, we was gradually increasing the CO2 levels, but here in A by Ci, A by Q curves, we are gradually increasing the light intensity. So by using this one, we could able to quantify 3 different parameters. First one is saturated photosynthetic rates, then apparent quantum yield, then dark respiration. So these are the 3 major important parameters, so by which we could always assess the efficiency of photosynthesis in different types of crops and different types of circumstances here. I hope you got this point. Yes or no? Yeah. Which one? Which one? This one? So which one? So at what light intensity the plants are getting saturated? PAR, yeah. Yeah. This one. So suppose you just see the 2 different graphs here. So one graph here is saturating at lower intensity and lower light levels, and another graph is showing the saturating at higher light intensity. So which one is performing better here? This black one is performing better as compared to this one. Yes or no? So here, this how I think we could always be able to perform the different things here. So people try to investigate the crop responses under changing climate. So people find, I think, the moment when we start exposing the plants toward this, I think, simulated levels of carbon dioxide around 2050. What I told here, by the year 2050, I think we are anticipating the CO2 levels could be around 550 parts per million. Yes or no? So people start doing this, anticipating. People start simulating this CO2 levels by different concentration. So I think the moments when people expose to high CO2 levels, they suddenly observed that the significant rise in photosynthetic efficiency around 50 to 60%. 50 to 60%. Then they also observed diminished somatal conductance. Then they also observed this significantly rise in water use efficiency. Then they also observed regarding about this increasing biomass yield. I told very clearly the changes in biomass or the changes in crop productivity is directly proportional to the photosynthetic efficiency. Photosynthetic efficiency. So more photosynthetic efficiency, more biomass, more crop productivity. Yes or no? So here, then again next one. So the augmented photosynthetic nitrogen use efficiency. So these all are the different ways I think plant could able to perform the different consequences here. So only positive response. They don't observe any negative response. See here, they also observe some different negative responses. I think you must have seen that our forefathers, our grandfathers are more healthy as compared to us. Yes or no? Because they were eating very quality food. So we are moving towards this modernization of civilization. We are not having a quality food. Rather, we are having some different types of foods. Yes or no? So, which are causing a lot of serious health complication because the changing climate is always induces this nutrient dilution process. Nutrient dilution. So many vitamin deficiencies. So many vitamin deficiencies and mineral deficiencies. Again, next one. So the other major implications of this climate change is we don't know how this the abiotic stress factors and biotic stress factors are going to affect the plant community, plant productivity under changing climate. So some studies are showing that elevated CO2 is ameliorating. Some are showing that there would be no change. Some are showing that I think even the changing climate even aggravates the effects of these abiotic stress factors. Now, so the other phenomena column as I think the long-term growth of these plants under changing climate or elevated CO2 induce a specific phenomena known as the photosynthetic acclimation. Photosynthetic acclimation. So what do you mean by photosynthetic acclimation? I told very clearly here. So the adjustment of photosynthetic machinery towards a particular climatic condition. So this phenomena is always called as the photosynthetic acclimation. Now, so why the plants will always exhibit the photosynthetic acclimation? The major implications of the photosynthetic acclimation is, the first one is I think the altered C by N ratio. What do you mean by altered C by N ratio? Suppose we are giving so much of food to the individual. So the people used to take more food and they will try to build up his body in a random way. There is no proper incorporation. Suppose if you are giving a balanced diet to the person or any individual. So what's happening here? So when we are trying to giving balanced diet to the person, so they will take up all minerals, all nutrients, all proteins and all carbohydrates. So all fats, everything will be in optimum range and which could able to support the growth of individual in a proper way. So here, what is happening in changing climate under increasing atmospheric CO2 concentration? So plants try to use more carbon dioxide and in this fashion, plant try to invest more nitrogen. So over a period of time, the plant reaches a stage in which the foliar levels of nitrogens are reducing in elevated CO2 grown plants as compared to the ambient CO2 grown plants. So which brings out a simple phenomena known as this altered C by N ratio. What do you mean by so? There is a significantly high carbon content as compared to the nitrogen. So which will induces a simple due to this one because of this I think source to sink imbalance. What is source here? Leaf. Okay, so the moment carbon dioxide enter into the plant, so the carbon dioxide has to be converting into the sucrose. And sucrose has either sucrose or starch, whatever it may be here. Yes or no? So suppose the plant is unable to accommodate more sucrose or starch molecule. What's happening here? Because due to the limitations in the sink. What are sink here? Sink is nothing but roots, stem, branches. Yes or no? So these all are the zinc. Suppose the plants having more zinc, plants are having more greater root, and there will be no rooting limitation. What's happening here? The plant tried to continuously, which one? Regarding about this export to the photosynthesis to the respective zinc components here. So I think what's happening, so whatever the amount we are eating, whatever the amount they are sequestering, and it will be mobilized toward the respective zinc components here. The moment once the source to zinc imbalance has been taken place, so which will eventually causes, I think the phenomena called as photosynthetic acclimation. See here. Now, so how do we recognize this? The plant is exhibiting the photosynthetic acclimation. The most important thing is that which one? I think the increased starch to sucrose ratio. So what do you mean by starch to sucrose ratio here? What is exporting sugar? Sucrose. The moment there is limiting in zinc, so sucrose again can be reconverting into the starch. The moment starch is happening, when there is a high level of starch, so the high level of starch again inhibit the photosynthesis. Yes or no? Next again, reduced ADP ratio. Again next one, decreased NADPH. Next one, diminished ribosco carboxylation, RBP regeneration, and AMAX again reduced photosynthetic efficiency. So these all are the different signs by which I think we could able to manifest, conclude, I think the plants are attaining which one? Regarding about I think the photosynthetic acclimation. Now, so these all are the I think our experimental facility. So we used to simulate the CO2 levels of 2050 year in the University of Hyderabad during my PhD. So call them as I think open top chambers. Because the top of the chambers I think were open and try to maintain the almost natural environmental condition except CO2 level. So we try to simulate, we try to pump I think the 550 CO2 concentration into the chambers, and we try to investigate how the 550 ppm CO2 is influencing the productivity of different types of crops. Now, so here I think so this is the CO2 analyzer, I think, which will control the flow of carbon dioxide into the chambers. So the moment once the chamber has full of 550, it will stop pumping up the CO2 here. So these are the I think CO2 chambers through which I think we used to pump the CO2 into the inside the chambers and all these things. So I think now, so these are the simple basic experimental approaches here. So during my PhD, so I worked on this simple model plant known as mulberry. So why we selected the mulberry as a model plant? Because these are the faster growth, wide genetic diversity, I think semi-deciduous, and another thing is high responsive to CoPC. Again, next one, it has a lot of medical and ethnobotanical values, and now the mulberry is considered as a which one? Regarding about the potential bioenergy tree species. So these all are the different things I think we made us to stick to the mulberry plant. Another thing is, most important thing is, the mulberry cultivation is happening in India. Nearly 2 lakhs hectares are cultivating the mulberry in India. So that's why we need to, it's highly imperative to check how this mulberry is going to perform under future climatic condition. Now, I think these all are the most different types of genotypes what we collected from all over the 5 different countries. I think from which one? Regarding about the type, India, Bangladesh, Myanmar, Thailand, and Papua New Guinea. And so this all will be collected, and we establish a mini farm in our area in the University of Hyderabad garden. So I think so what could you observe in this from this figure? What could you figure out from this one? See here, I think these are the different types of which one? Regarding about that, I think mulberry genotypes. So this selection 1, Thailand, Mysore local, selection 165, Johari Paki, and triploid. So these all are the different types of genotypes. So what did you figure it out? What can you figure out from this picture? So I think you could see here, there is a significant difference in stem girth and plant height, leaf structure, leaf quality, leaf color, and all are different parameters, even though they belongs to same species. So people try to observe there is a change in genetic variation. There is a change in genetic variation in performance of photosynthesis. Though even though they all components that belongs to the same species, but what happened here? So they could be able to observe certain changes in the, I think, even genetic changes here. So here, the moment one, the change in photosynthetic efficiency, so which also reflected in the form of yield, biomass yield. So this is how I think people try to figure it out, how the plant is able to exhibit a respect to, I think, a specific photosynthetic process here. Now, so I think I'm going to show this one, I think, very important phenomena. So these are the basic morphological changes, what we observed during this mulberry. I think what you figure it out from this, I think you just see here. So do you observe any difference from this? These are the plants which showed morphological changes grown under 550 ppm, and these are the plants which grown under the 400 ppm, 400 ambient CO2. What did you observe? Any difference here? Color of leaf, okay. Apart from that, any? Plant height, number of branches, increased, right? Yes or no? Yeah. The moment we expose the mulberry to 550 ppm, there was significant rise in photosynthetic efficiency, which could reflected in the form of changes in biomass. Plant height was increased, number of branches, number of leaves, stem grid and roots, whatever it may. I think I'm going to show that. So then another important thing is we are going to evaluate how this elevated CO2 is interacting with abiotic stress factors, so-called drought. So what is happening here? So here, these are the plants which were grown under elevated CO2, and these plants could show the morphological changes. So after 30 days of complete water withholding, we have not given any water. To the, I think, this mulberry plants. I think so to check this, how this plant is going to perform even under the drought condition, because the drought episodes are more pronounced under changing climatic condition. So we would like to emphasize, we would like to investigate that how these are going to happen in order to, I think, so we understood that, I think, the elevated CO2. So what did you observe in this? There is any changes in this? Which plants are more healthy here? Ambients are more healthy. See, here more leaf is fallen here. Here all leaves are completely wilted here. What about here? Leaves are still healthy here. Where? Yellowing. It's okay, but here completely dried, right? Here yellowing is there, but here completely dried and they fall on so much of leaf here. Yes or no? So what we observed that, I think, the elevated CO2 is try to ameliorate, try to nullify, try to which one regarding mitigate the impact of drought in, I think, which one regarding about, I think, the mulberry here, Now, so this is the stomatal behavior. So what is happening in case of mulberry plants? So when we expose to 550 ppm. So what did you figure it out from this figure? These are the ambient and these are the elevated. I think in case of ambient plants, more stomata open. The stomatal aperture is also more. As compared to the elevated CO2 grown plants. Elevated CO2 grown plant means the stomatal adaptation has been taken place in the plants which are growing under elevated CO2 conditions here. Then, so these are the photosynthetic physiology, what we measured through IRGA, what I told here previously. So I think the IRGA can be used to track the changes in photosynthetic efficiency. So this is called which one? I think the photosynthetic rates. So elevated CO2 grown plant showed higher photosynthetic rate without drought as compared to ambient CO2 grown plant, but the drought imposition reduces photosynthetic efficiency, but the efficiency was reduced more in case of ambient CO2 grown plant as compared to the elevated CO2 grown plant. Again, see here, reduced stomatal conductance. I think I told very clearly. So the plants which are grown elevated CO2 try to maintain higher intercellular CO2 concentration. I think lesser transpiration in elevated CO2 grown plants, then followed by the, I think, more water use efficiency. And so these all are the different parameters which could always take in place to assess the plant performance under changing climate. Assess the plant under the changing climate here. So now, so these are the, I think, FU by FM. What is FU by FM? Maximum quantum yield. Okay, so this is the changes in photosystem to efficiency, then followed by the yield, non-photochemical quenching, and relative water content. What is relative water content here? So in order to assess the relative water content, we have to check this leaf water status. See here, there is not much change in the control condition, but when we are giving the drought, what's happening here? So I think the elevated CO2 grown plants are trying to retain more water, because there is a less transpiration rates. As compared to the ambient CO2 grown plant. I think so. These are the different curves. This is the A by Ci curve. A by Q curves. I think increasing photon flux density and increasing, I think, with respect to CO2 assimilation rate. So plants which were growing under elevated CO2 were tend to having higher photosynthetic rates even under both well-watered as well as drought condition, as compared to the ambient CO2 grown plant, which were further reflected in the form of which one? Regarding about this the apparent quantum efficiency, light compensation point, and A saturated, which clearly indicating that increasing atmospheric CO2 levels are try to minimize the photorespiration, and which eventually causing this more photosynthetic efficiency. I think this is the very important fast chlorophyll, OJIP chlorophyll fluorescent transient. I think so. It will always track the flow of electron from photosystem 2 to, I think, photosystem 1, including from the water splitting complex, efficiency of the water splitting complex. So which can be divided different regions. O region, I think this is the this part will emphasis on OJ region, and this part will emphasize on JI. This part will emphasize on, I think, IP region. So which will be always tells about the flow of electron, how the electron flow has been taken place into the in case of different consequences here. Now, so these are the results what we observe. See here, I think these are the raw chlorophyll fluorescent transients, and these are the which one regarding about this, I think the normalized things. I think I don't want going deep in this, because I think you may get again confused here. So I will just summarize these consequences here, and this normalization we did for this the observing of this a specific type of band known as, I think, L band. So what is the major concern about L band? Why do you look for this L band? So see, these are the well-watered plants. We don't observe any L band. But the moment once you try to impose the drought, so then which will always able to produce a unique peculiar band known as L band. This L band will majorly emphasize the connections between light harvesting complex as well as the photosystem 2. Suppose there is a rise in amplitude of L band. There is a high, suppose see here, which one here? This is the pink one. So pink means what's happening? More L band here. So these are ambient CO2 grown plants with drought. So if there is this is a inversely proportional. More L band, less connection. Less L band, more connection, more stability. Yes or no? So here, so what's happening here? So there is a rise in amplitude of L band in ambient CO2 grown plant, which clearly indicates that I think this plant doesn't have proper stability. There is a destabilization of which one regarding about that, I think the light harvesting complex as well as this the photosystem 2 complex. Then we also tracked for the another type of band known as which one regarding about the K band. So the K band will majorly emphasize this the efficiency of water splitting complex. See here, the moment once we observe this rise in amplitude of water split K band, so which clearly indicates that inefficiency of oxygen evolving complex. So here, so what's happening? So that's why I think so this ambient CO2 grown plants doesn't have the proper oxygen evolving inefficiency of this I think the oxygen evolving complex. So these are the very important parameters we could able to assess the efficiency and stacking and stabilization of this I think chloroplast membrane, and so which eventually causes the simple which one deformalities regarding about in case of the plant performance here. Then the changes in chlorophyll fluorescent transients were further translated into Gyp test parameters. So there are so many Gyp test parameters here. I don't want going deep into that. I think I specifically focus on this which one regarding about some important parameters which tells about the photochemical quenching as well as non-photochemical quenching. So what do you mean by photochemical quenching? And what do you mean by non-photochemical quenching? Suppose one plant is there here, two plants are there. One plant could able to take up 80% of light, and 20% light is emitted in the form of heat. Then another plant could take up this 60% of light and could emit in the form of light, 40% in the form of light, heat energy. So which one is more efficient? Plant A. So because this plant could able to exhibit more photochemical quenching, so that's why the moment one it has started performing more photochemical quenching, and the flow of electrons will also more. Okay, so here now, so these are the more important parameters, I think Fe by Fm, Fe by F0, electron transport for cross section and performance index. So these are the parameters which tells about that how this the plant is going to show, this plant is going to exhibit the process called as I think the photochemical quenching. So what we observed that at both elevated CO2 grown plants at well-watered as well as drought condition, they showed the rise in the parameters like Fe by Fm, Fe by F0, electron transport chain and performance index, and with concomitant reduction in the other parameters which related to non-photochemical quenching, which one regarding about I think F0 by Fm, Kn, dissipation per reaction center, dissipation per cross section, so which clearly indicating that the plants which were grown under future atmospheric CO2 concentrations are having more photochemical quenching in case of mulberry, but it is not applicable to all other plant species. I told very clearly the plant responses are highly specific, and even the plants they belong to same species are going to show some difference in photosynthetic efficiency, and we can't apply the same principles, we can't apply the same response toward this another type of plant. Suppose we can't compare the rice plant performance with mulberry plant performance. You can't compare the mulberry plant performance with another X plant. So here, so these are the plant responses are highly unique, and that's why we are trying to screen very important and very potential potential crops. So here, so which clearly indicating that the elevated CO2 improve the performance of these plants, improve the performance of photosynthesis in case of mulberry plant, so which will eventually reflected in the form of this increasing biomass yield. You already seen. Yes or no? Now, I think this is another story, very important story here. I have been telling that there is a less membrane destabilization, means there are more membranes are more stable, more efficient, and all these things is happening in case of this elevated CO2 grown plants. Now, I am trying to correlate with this I think antioxidant metabolism. So what is antioxidant metabolism? Do you have any idea about antioxidant metabolism here? I think it is ubiquitous. Just look at animal cell, look at plant cell, look at algal, anywhere. Antioxidant metabolism is universal, ubiquitous. Without this, I think there will be no existence of any type of component. Yes or no? The moment we start feeling stress towards something, our system try to combat that stress through a signaling process called as ROA signaling, reactive oxygen species. Suppose you feel more stress. Whenever you feel more stress, our cells try to produce more ROAs. What are the example of ROAs? Oxygen radicals, hydroxy radicals, H2O2, singlet oxygen, doublet oxygen, whatever it may be here. So, which will eventually degrade the cell structure and function. How these ROS are going to degrade here? So here, the ROS will always able to induce oxidative stress, so which will oxidize lipids, amino acids, proteins, nucleic acid pigments, and which will eventually cause this cellular damage, and finally the cell will subject to death. That's why the cell has defense mechanism. The cell has developed some defense mechanism to combat, to sequester these reactive oxygen species. Suppose the production of ROAs is equals to the sequestration of ROAs, then there will be no problem. Plants will happily survive. The balance has moved towards which one, that is more ROAs. The balance has been shifted towards more ROAs, then which induces oxidative stress and which will eventually cause this the plant cellular death. Are you clear? So more ROAs, more oxidative stress, more stress effects. Less ROAs because efficient antioxidant metabolism, and less oxidative stress, and finally plant will survive happily. You don't have any problem with that. So we want to track that, how this antioxidant metabolism is going to be changed in case of plant cell, Now, So we have seen and we perform the, I think, different types of activity under elevated CO2 with respect to drought as well as well-watered plants. Surprisingly, we observed that, see here, the dotted lines are ambient CO2 grown plants, and the thick lines were elevated CO2 grown plants, and we did not observe any elevation. We did not observe any elevation in the antioxidant metabolism until the 30 days. What this indicates that? What this indicates that? Why plants are not coping up? Whenever you feel hungry, then you will eat, right? You don't feel hungry, you don't eat, na? Yes or no? Because the plants are not feeling stress. Then why do they able to produce more amount of antioxidants? They are more healthy. Are you clear? So the moment the plants are more healthy, then they are saving their energy. They are storing their energy. They don't want exhibit, they don't want perform the unnecessary activity. So here, but the ambient CO2 grown plants are feeling more stress, and which eventually causes, I think, so what's happened? There is a surge in antioxidant metabolism. And until the 15 days of that, but the ambient CO2 grown plants could not able to handle this ROS, and which eventually causes this death of plant cell through the oxidative stress. So here, this is how I think what's happening here. But here, these are the ambient CO2 grown plants, even they could able to handle the stress even after 30 days of the stress. After stress. So now what's happening? So this is happening with all different types of enzymes here. So here, there are different enzymes are there: superoxide dismutase, ascorbate peroxidase, catalase, glutathione reductase, GPX, MDHCR. So these all are the different enzymes they could take what in the process of antioxidant metabolism. So that which could able to sequester the ROS and finally could able to neutralize this ROS component. Then we have seen similar result in case of transcriptomic analysis. So we perform the QRT-PCR analysis, that is the gene expression studies. So the gene expression studies also we have also observed the similar changes. There was a significant rise in transcript levels of different antioxidant enzymes in case of ambient CO2 grown plant, even under well-watered conditions. Then the similar trend was observed even after 15 days, but after 30 days, the ambient CO2 grown plants could not able to handle the stress, and finally, so they lowered this the activity of all type of enzyme because the cells were died. So this is happening with regarding about, I think, the changes in the gene expression of different types of enzymes. Then I think these all are the non-enzymatic antioxidants: ascorbic acid, total phenolic content. I think the proline. So do you have any idea about proline role? Have you heard about proline? What is proline? Proline is amino acid. Yes or no? So what is the role of proline in abiotic stress? The proline, the quantity of proline will always tells us the stress level, stress intensity. More proline, more stress. Less proline, less stress. Yes or no? See here, now what's happening here? So here, ambient CO2 grown plant showed more proline quantity here. Indicates that they are feeling more stress, which could be reflected in the form of H2O2 levels. So they are accumulating more H2O2 because they could not able to sequester ROS. Inefficient antioxidant metabolism. Though finally, so I think another important for MDA. What do you mean by MDA? So what is the indication of MDA? Melandialdehyde. So this is the parameter which will always tell us about lipid peroxidation. So I think you know very well all members are made up of lipids. So the moment there is a rise in MDA quantity, which clearly indicating that the lipids are more subject to oxidation. So this is why the plants are grown under elevated CO2 showed more healthier as compared to ambient CO2 grown plant, and the destabilization has been taken place more in ambient CO2 grown plants as compared to the elevated CO2 grown plants. Are you clear? So these all are the different consequences what we observed in case of this consequences. So here, I think these all are the different biomass. I think so. These are the morphological differences. I think DAT stands for days after treatment. Then followed by the here. This is 90 days, and these are the morphological changes after 360 days after 1 year. And so this is the different types of biomass what we observed after 1 year when we harvested the plant. So which were grown under which one? Under elevated CO2 grown plants, and as well as the ambient CO2 grown plant. I think you must have asked about that. What is SRC? Do you mean what is SRC here? I think your short rotation COPC. What do you mean by COPC? Suppose outside one tree is there, neem tree is there. If you cut down the plant, to grow up, it will take years. Okay, but when you cut down the plants for mulberry, it will resprout within a span of 3 months. So we used to harvest the plant for every 3 months, and we used to generate the biomass for every 3 months. And because our motto is able to produce the more biomass for production of more energy, more bioenergy. So this is how what is happening that the SRC1, SRC2, SRC3, and SRC4 yearly we used to harvest the mulberry plants for 4 times. 4 times, and we used to generate a lot of biomass, and this biomass can be able to reflect. See here, this is the biomass what we generated in elevated CO2 condition after 1 year, and this is ambient CO2 grown plant. We just see here the number of main primary branches, and here these are the major differences regarding about that. I think the plants grown under elevated as well as ambient CO2 grown plant, and this is the root biomass what we observe. And we calculated carbon sequestration. I think the ambient CO2, elevated CO2 grown plants almost, I think, see here, this is the plant biomass after 1 year, nearly 20 kg in elevated, and this is the, I think, 10 kg. How much difference? 50%. Okay, so here we calculated CO2 concentration, and we also calculated the amount of CO2 sequestered by this dry biomass plant. Then, so finally, we could able to extrapolate towards which one? Regarding about that, I think, toward this per hectare. I think, so these are the plants. I think similar things is happening regarding about that. I think, so these are the plants even after 3 years. So we continued our experiment for 3 years. That was first 1 year, and we prolonged this experiment for 3 years. So we would like to know how much how the plant is going to tolerate for this, how long it can tolerate, how long it can perform this photosynthesis. Still, we could able to see. I think you can see clearly visible this difference here. I think this ambient CO2 grown plants and elevated CO2 grown plant, still it is performing better per photosynthetic efficiency after 3 years. So after 3 years means how many harvests we have done? We did how many harvests? 12 times. We harvested the 12 times. So again, we calculated this biomass. I think after 3 years, the biomass could be around nearly 85 kgs per individual plant, as compared to the, I think, 35 around nearly in the elevated CO2 grown plant. And the amount of CO2 sequestered by this when we extrapolated towards per hectare, nearly around the 400 megagrams per, I think, tons, 400 tons of CO2 has been sequestered by this mulberry per hectare. So what we are indicating that I think these plants are more efficient. So, but what we observed surprisingly that, so when we track the photosynthetic efficiency, changes in photosynthetic efficiency through A by Ci curve, so I think what I told here, the rise in amplitude of these graphs. So there were, after 6 months, the plants showed better photosynthetic rates. I think we don't have any observance. We didn't observe any acclimation, but the trend was changing when we are moving toward this. When the years were progressing. See here, I think after 1 year, I think the elevated CO2 grown plants could not able to exhibit a proper, I think, a proportionate photosynthesis as compared to the ambient CO2 grown plants. Again, so this is happening after 3 years, and so that's why. So when years were increasing, so which eventually causes this the down regulation of photosynthesis. I think we had a lot of I think experiment not to confirm all these things. So I think the ultimate one is, I think there is a significant dilution of nitrogen. So I think so this is Vc max, J max, and A max. So it could be further reflected as we seen here. So I think so there was a higher in 6 months in elevated CO2 grown plant, followed by the, I think, reduction in, I think, after 1 year and 3 years. Again, so to confirm this, photosynthetic acclimation, we perform this comparative proteomic analysis, and in this we have seen the different types of proteins which were showed some differential expression, and which are related to cell signaling, cell wall biogenesis, energy metabolism, fatty acid biosynthesis, nitrogen metabolism, photosynthesis and protein metabolism, and different protein. So I think nearly we showed 70 different proteins. 70 proteins are showing the differential expression. Now, but we are more focused about photosynthetic protein. We wanted to correlate these changes in A by CI curves with the protein expression. So what we did, so we have done this comparative proteomics, and we identified, different types of proteins which are related to photosynthesis are showing this down regulation here. So this is the Rubisco large subunit showed down regulation. There is a significant lower in elevated CO2 condition as compared to ambient CO2 grown plant. Again, next one, the glutamine synthetase, the most important enzyme which is take part in nitrogen metabolism, is higher in case of elevated CO2 grown plants. Rubisco active is also very high, and finally, some other proteins like oxygen evolving enhancer protein, pseudoepitose 1,7 bisphosphatase, and phosphoribullokinase. So these all are the different proteins, and they showed this down regulation. And so which is having some important consequences. So which clearly tells us that so the mulberry plant which are grown at elevated CO2 after 3 years starts showing this down regulation. Process here now. So with this, I could conclude. So the climate change is more prominent. We should be very cautious and very conscious about this crop productivity, and which are going to have a lot of negative impact on our society as well as human health, and that's why we should be very careful regarding about this thing compared to all these things here. So I would like to congratulate this with this all organizing committee, and I would like to say about my teacher, Professor Rajgopal sir. He's our teacher. So though we are not direct student of Rajgopal sir, but we admire him, we love him, and we respect him a lot. And he's a very good human being. He's helpful to everybody. Suppose if you want to work under him, you just write a mail. He will accept you, come and work with me. You don't have any problem with that. Such a nice person, such a wonderful human being. Okay, so this would I will conclude my session. Queries? Yeah, any queries, please let me know. I'm happy to answer your queries.
Sir,
yeah,
I have one question.
Yeah.
So is there any effect of ambient and elevated CO2 in silkworm production of the silk?
Yeah, because that would be another PhD work. Huh? So we didn't check it. We didn't use that leaves for how this actually we would have any collaboration with somebody else would have tried that, but unfortunately we didn't do that actually. Definitely there would be some effect over that.
Maybe you are thinking positive or negative thinkings are there.
So because this plant is performing better, so I am hoping that positive response. So but depends upon the plant age also, depending upon nutritional status. If you are starving the plants, definitely we will be having some negative impact. If you don't starve the plants and we are feeding them happily, then plant could be able to perform better, and it might be having some positive impact over that.
Sir, mulberry plants for the biomass growing. So water is requirement is needed more now actually.
Yeah, that's what. So not needed much actually. We used to give water for every 10-15 days like that in our field. But it's a water demanding plant, no doubt about it. Because these are fast growing tree species, right? Mm-hmm. So we have to always give some. But in some area, this plant is growing in a right-fed manner through rain-fed manner also.
Thank you, sir.
Any?
So I have a question about the quantification of
ROS?
Calorimetric?
Calorimetric, yeah. Yeah, calorimetric.
What about enzymatic activity?
We did calorimetry only. Yeah, everything calorimetry only. Not spectrophotometry, not calorimetry.
Full spectrum?
Yeah, full spectrum. So many protocols are there in literature. We can go through it, no problem. This RT-PCR we did through, real time. And the remaining all are calorimetry, spectrophotometry. Any queries? I'm happy to answer your queries.
Anybody is having any questions?
Come.
Sir, the increase in carbon dioxide levels, are they good for the plant?
Yeah, see, Increasing carbon dioxide level is good to some extent. Okay, but the way we are pacing now, I think it is going beyond our limitations. For some time, increasing atmospheric CO2 to some extent it is beneficial. But I think I told here, the moment once a rise in CO2 level, there is a rise in temperature also. There is a rise in prevalence of more droughts. I think there is lots of variation, lots of changes in climate. So which eventually all combine together, and instead of having positive impact, these plants could able to, I think climate showing more negative impact. When we are integrating individually, it's having positive impact. But when we are integrating all other factors, it's more effective. Clear?
Because you also said that over the 3 years, the nitrogen content
Yeah.
is diluted.
Yeah.
So that may also have effect on the plant.
Yes. Because of that, because the nitrogen dilution may not. That's what he was asking question that. So how these plants are going to fetch? Suppose if you use these leaves to raise the silkworm, so how about their quality of silkworm? Because definitely the moment one you see that, because we have already seen that, there is a lot of nitrogen dilution. There is only increasing biomass. There is only increasing quantity, but there is no quality. We need quality, right? So this how I think it will be cumulatively affecting this total overall growth and performance. Are you clear? Yes.
There are no questions, we need to thank Professor Dr. Madan for wonderful talk.
Thank you, Venkat. Thank you so much. And